Welcome everyone to today's webinar, which will cover the Analytical Technologies for Cell Therapies chapter of ARM's A-Cell Best Practices case study. My name is Michael Lemke. I'm the Vice President of Science and Industry Affairs at the Alliance for Regenerative Medicine. I'm going to just make some brief introductory remarks before I turn over to today's presenter. Hopefully you know us, but in case you don't, the Alliance for Regenerative Medicine, or ARM for short, is the global voice of the cell and gene therapy sector. We are a not-for-profit advocacy group. Our members are organizations, currently in excess of 475 of them, primarily companies who are involved in the development of cell and gene therapies, but also technology providers, CDMOs and CMOs, medical and research centers who are the source of much of the innovation in this field, patient advocacy groups, and other stakeholders. We work to convene the sector, and you're going to see a partial result of that today. We convene in many ways through large events, including our upcoming cell and gene meeting on the Med and our cell and gene meeting on the Mesa in October in California. But we also pull together subject matter experts from our member companies to work on best practices. And this is really what the A-Cell Guide is. I'll tell you a little bit more about that. We've identified with our members the need for best practices, specifically quality by design principles in the manufacture of cell and gene therapies. As I'm sure you all know and are experiencing, manufacturing is a rate limiting step in the commercialization of regenerative medicines. And until we can address that, it's going to affect patient access to these important therapies. Why is this so much of a pronounced issue right now? It's really because we're seeing rapid scale up in the industry and more phase 3 and registrational studies. For example, there were 6 new therapies approved last year. We're expecting to see as many as 15 approved this year in the United States and Europe. So we're seeing more products. That's good news, but we're also, as this occurs, seeing more products being held up at late stage due to CMC issues. I like to think of process understanding as having 4 critical elements. 2 of them are experience of people in the field, and also our knowledge about technology and our knowledge about the products and the processes. Those 2 things can advance sometimes in a competitive way and be a source of competitive advantage. That's all good and good for the field, I think. But there are also places, because there are limited resources, no one company can solve all problems or address every issues. So there is a need for cooperation and collaboration in a pre-competitive sense to address best practices and, in some cases, standardization. And without those things, it's my belief that the field really won't advance. You need all 4 of these things. Today, we're going to talk about a specific example of best practices. And this is the A-Cell case study, which is a collaborative case study describing the implementation of quality by design principles in cell therapy manufacture. In this case, the specific example is a hypothetical autologous CAR T product. This A-Cell follows the example of another case study developed by ARM called A-Gene, which describes quality by design principles used in the manufacture of AAV vector. And we were originally inspired by AMAB, which was not an ARM project, but a collaborative industry project going back some time in the monoclonal antibody industry, which really inspired us to do something similar for cell and gene therapy. The content of A-Cell was developed with the input of subject matter experts from over 30 participating ARM member companies. We led and managed this process, and we were fortunate to have funding to support it from Nimble. The document is open access, and you can see here in the link down at the bottom where you can download it from our webpage. The contents of A-Cell are extremely comprehensive, as you can see here, really walking through regulatory considerations, how you generate quality target product profile, how to identify critical quality attributes using risk analysis and empirical methods, and exhaustive description of the process. Today, we're going to focus on one very important aspect, which in ACell is Chapter 9, Analytical Technologies for Cell Therapies. This is just a quick word about our webinar series. So you're here today on March 21st. This is the fourth webinar we've held on ACell. We currently have 2 more planned, one on April 5th on ancillary materials and one on May 2nd on control strategy. And if you follow the link on this page, which we'll also make available after the webinar, you can register for these for free if you aren't our member. And we put recordings of these webinars on our website afterwards for people to refer back to later. With that, I would like to introduce our expert speaker for today. It's been my pleasure during the time that I've been at ARM to work with Rupa Pike on the ACell project and on other initiatives. Rupa is currently the Senior Director of Technical Affairs and Advanced Therapies at Thermo Fisher Scientific. She has over 2 decades of experience, academic and industry experience in regenerative medicine. She's built cell therapy programs, led GMP manufacturing operations. She has driven strategic partnerships and importantly, designed training programs for workforce development. So obviously very qualified for the task at hand today. Rupa, thank you for joining us and welcome. And with that, I will turn it over to you. Just a logistical thing for the audience. We do have the Q&A window active, so I will be managing that. Please feel free to post your questions at any time, and we will have time at the end to get to your questions.
Thank you, Mike, for the introduction and also for setting the stage for the entire ACell project. Hello, everyone, and thanks for joining. So as Mike mentioned, today's webinar is focused on analytical technologies for cell-based therapies. This webinar is designed based on the ACell book chapter that Mike referred to. Next slide, Mike. So before I start, I would like to thank everybody who participated in the chapter. And as you probably already figured out, this was truly a team effort. So I'd like to thank, first of all, Taylor Zhang, who was my co-lead. I would also like to thank Mike Sadik, Joseph Lanning, and Anya Fritsch. And also a big thanks to the ARM team, Mike, who has joined us today as a moderator, and Josephine and Amar, also from the ARM team. Next slide. So just to set the stage, here's the agenda for today's webinar. So I'll start with some background. I will speak about analytical assay development strategies, touch base on types of analytical assays. Then I will discuss approaches to improving assays. I will review process analytical technologies or PAT or in-process assays, which are very important. And then spend some time on potency assay dilemma, which I believe is a hot topic. Then I will discuss future advancements, which are very important in the analytical assay development space, and then close with a summary. Next. So just to set the background, there's no doubt that cell therapies are here to stay, since they offer life-changing treatment options for patients who have no other recourse. As you know, in the hematological malignancy section for the approved products, there are a large number of patients that are relapsed or refractory to traditional medications, and they have seen success with these patients, starting with experimental cell-based therapies. Autologous therapies, as you already know, have enjoyed commercial success with 6 products on the market. And last year, we saw the first approval of Atara's allogeneic therapy in EU called Abvelo, which is very, very exciting. Now, these advancements are wonderful, but they have also fueled the need to develop robust and well-characterized analytical methods that are critical for demonstrating safety, identity, purity, strength, and potency for the final product. Since cell-based therapies are unique and the final product is comprised of living cells, the complexities associated with cell therapy manufacturing require a broad set of analytical tools. Analytical methods are integral to the success of the final product, since they play a critical role in assessing raw materials, as you see on the slide, the manufacturing process itself, intermediates that are generated during the manufacturing process, and of course, the final product. It is critical to develop and employ analytical assays as early as possible in the process development stage. Why? Because they provide insights into the attributes of cells throughout the manufacturing process, and they help enable process changes and also offer a better control of the final product. Next slide, please. So in this slide, I would like to make a connection between critical quality attributes and analytical assays. Those of you who've attended the previous webinar and read the chapter, you know that there's a lot of very good information in that chapter about critical quality attributes. Critical quality attributes, or CQAs, are the physical, chemical, biological, or microbiological properties of the cell product that should be within an appropriate limit, range, or distribution to ensure the desired product quality. This is the definition for critical quality attributes. And as you can infer from that, critical quality attributes and product quality are intricately connected, and analytical methods are used to assess critical quality attributes. So that is the connection between those two. There's a general consensus that analytical assays should be designed during early process development stage and subsequently refined to ensure product quality and optimal CQAs. I do want to touch base on a little bit of a provocative topic. We have all heard the phrase growing up in the cell therapy space that in cell therapy, the process is the product. In fact, I have used that statement many times too. Well, many knowledge and thought leaders are now saying that it is time to stop using this phrase. They believe that the product is in fact the product, and the process does not need to be the product. The product can stand on its own with better characterization, with new analytical tools and methods that incorporate omics, deep sequencing, and other advanced technologies. There is also a lot of chatter about biosimilars in the cell therapy space, as many of you who attended advanced therapies would have heard speakers talk about it. While that is far away, today we still define the product by controlling the process. But once we have a clear understanding of the true critical quality attributes and assays that are sensitive enough to evaluate these, the field can then begin to advance to a quality by design approach where the process operates within a range and the CQAs and not the process truly define the product. And I would like to talk about this a little bit in our QA time. Next slide, please. So this slide describes the approaches to analytical assay development. Many cell therapy developers, as you probably already know, use phase-appropriate or fit-for-purpose assay development approach. And the diagram shown here shows the progression of assays from the preclinical to the post-licensure stage. And the diagram, as you walk through from left to right, shows that early qualification can begin during phase 1 to 2A, while late phase validation comes at phase 3 and BLA. If you look at the boxes underneath, it states that phase qualification includes limited repetition using the power of n, limited parameters, and wider acceptance criteria. Late phase or phase appropriate validation includes full number repetition using the strength of numbers, all parameters for the assay, a more stringent acceptance criteria based on data that has already been generated, and robustness of testing. And the last one on the right side, the post-licensure activities, they require data trending and method performance evaluations, method evaluations and comparability, and revalidation as and when necessary. It is important to note that risk assessment exercises are needed to identify the critical quality attributes and determine the timing of qualification and validation for the corresponding release assays. Next slide, please. So here are some of the fundamentals on developing strategies for analytical assays. In case of cell therapies, it is vital to develop analytical assays that build upon the relationship between donors or patients starting material, donors in case of allogenic, patients in case of autologous, and then raw materials, variables such as media supplements, cytokines, growth factors, extracellular and matrix substrates, product variables that include expected mechanism of action, understanding of the cell phenotype, marker expression of the cells, proliferation profiles, metabolic profiles, apoptosis, exhaustion, all of these criteria. And then process variables that include media perfusion, exchange rate, feeding regime, pH, dissolved oxygen, etc. This is to ensure that product quality is maximized and CQAs are maintained within the expected range throughout the life cycle of the product. Continuous evaluation of assays and streamlining select assays for good manufacturing practice, also known as GMP, ensures standardization and reproducibility. It also reduces deviations, increases the quality, and consistently meets the release criteria. And lastly, it also ensures a smooth transition from clinical to commercial manufacturing. Next slide, please. So this brings us to the topic of assay development, qualification, and validation. And many of the cell therapy developers have gone through all these stages, or some of them have just gone through the development and qualification stages, depending on where they are in the process. It goes without saying that assays need to be developed as early as possible. We have heard that from many sponsors, many white papers, many thought-provoking articles, as well as from the regulatory agencies. So during process development, qualified assays should be continuously subjected to reoptimization or requalification if process changes are needed to maintain suitable performance. This is an important task. It is laborious and it is tempting to cut corners. But I think that the reoptimization and requalification is necessary to have successful validation and also a successful GMP manufacturing strategy. High-risk assays that measure safety parameters, those would be mycoplasma, sterility, endotoxin, just to give examples, should be qualified and validated in a phase-appropriate manner. But now I have seen people say that maybe go to validation as soon as possible, if at all possible. And then when changing an assay, a risk assessment should be performed to determine how the assay change impacts evaluation of the cell product. Next slide, please. So this slide describes the types of analytical assays and the time points at which they are executed. As you can see in this slide, you can have assays that need to be in place for one starting material, and that could be anything ranging from an apheresis product, blood, bone marrow, cord blood, or cell lines in case of induced pluripotent stem cells or embryonic stem cells or dendritic cells for vaccines. Next one is the intermediate material, and for use in process analytics for intermediate materials. For final product, identity, purity, impurity, potency, and safety is critical. And these tests are also used ultimately to determine product stability. The second set of points is about the type of assays used in cell therapy, and the names are probably quite familiar to you. And they are listed as release assays, for information only, which are FIO assays, and mechanism of action, which is MOA assays. So a little bit about a couple of the top ones, and we will discuss the MOA assay with potency in the upcoming slides. So release assays are a panel of assays required to test the safety and in many cases, efficacy of the final product, even in the early stages. Once the product passes these tests, it is released for administration to the patient. Release criteria are predetermined in the CMC and already approved by the regulatory agencies. The sponsors and manufacturers may decide to use assays that are under development to evaluate product characteristics or method performance capabilities. Such assays are designated as FIO or for information only assays. And FIO assays, while not used for final product lot release testing, many believe that they're vital for understanding additional quality attributes for the cellular product and the mechanism of action, a window into understanding the mechanism of action. They're also helpful in gathering more information about the key steps of the manufacturing process or the final product. FIO assays can be tracked through process changes to reinforce comparability and help identify non-crucial characteristics. So an example would be determining time points in culture when cells stop producing a specific cytokine, even if that does not affect its target function in patients. So that is a really good example. FIO assays, as many people have stated, are indispensable as backup assays if there are problems validating existing release assays. For example, the existing assays are too variable because of the inherent variability of cells or starting material, supply shortage, or discontinuation of critical materials. Those are two good reasons. And lastly, in cell therapy, the MOA assays are tied to potency, which I am planning on covering in the upcoming slides. Next one, please. So here's the list of analytical assays used for cell characterization. And as you probably know, they all fall under 21 CFR, Code of Federal Regulations, Part 610. And as I have shown on this slide, they can be divided into safety, identity, purity, potency, and constituent materials. Safety, as you can see, includes sterility, pyrogen testing, mycoplasma, adventitious agents, vector copy number, and replication competent lentivirus. The last two apply to gene modified cell therapies using vectors, and the last one specifically using lentivirus. The next one is identity, and this is a specific test to distinguish the cells or the product from other products manufactured and tested at the same site. Purity is an assay that determines if the product is free of any extraneous materials. Potency, which is a very important test, is the measurement of biological function or activity of the cells, which is the final product. And constituent materials include ingredients, preservatives, diluents, adjuvants, excipients, and ancillary materials. Next slide. So now that we have covered the foundational aspect of analytical assays, I would like to focus the second part of the webinar on practical aspects and applications of these assays. So in the next two slides, I would like to talk more about some common pain points or challenges and approaches to overcome them. On this slide, you can see on the left box, the first is the mycoplasma assay, which is part of the safety panel. The 28-day USP63 or EP 2.6.7 test has been the gold standard for a very long time. But everyone is realizing that this may not be the ideal for cell therapy products. Why? Because the long testing cycles lead to delays in lot disposition and adversely affect patient outcomes. Also, generally, this test is outsourced. It involves shipping logistics, and it can get expensive. So what is the solution? The solution is to replace the 28-day mycoplasma test with a rapid qPCR-based test. This test can be easily performed in-house in 5+ hours, which enables same-day lot release if necessary. Its sensitivity meets or exceeds culture-based tests, and it can offer significant cost savings. And currently, there are companies that provide this qPCR-based test, and these are very commonly used by the FDA and many of the sponsors and manufacturers as well. The second example is sterility testing. The current gold standard compendial test for pharmaceutical sterility requires that the sample of the product be placed into aerobic and anaerobic recovery media for 14-day culture, which means that the test takes 14 days. This is also a problem here because long testing cycles lead to delays in lot disposition and also adversely affect patient outcomes. This test is also generally outsourced and involves I'm still on the previous slide, Mike, sorry. Thank you. So this test also is generally outsourced and it involves shipping logistics and also it could be expensive. So what is the solution for this? Sterility assays for microbial detection can be developed and validated in-house using state-of-the-art automated instrumentation. Couple of examples are bioMérieux's BacT-Alert 3D system and BD's BacTech system. These are very commonly used. The sensitivity of these systems meets or exceeds culture-based tests, as has been shown many times. And this type of testing can be conducted in-house for faster lot release, and it may offer some cost-saving measures. Next slide, please. So here are a few more examples that approaches we can take to overcome the pain points. The top one is example for in-process testing or sampling. So here the pain points are extensive sample plan requires a lot of cells. This can create a challenge in meeting the clinical dose, especially for autologous therapies where the starting material is very precious, could be very little, and having an extensive in-process testing plan can jeopardize the final number of cells required to make the clinical dose. So one of the solutions is to use an easy qPCR-based fast and sensitive assay that requires a small sample size, as little as 2 to 5 microliters. It detects low levels of protein with greater sensitivity than traditional methods like ELISA. It has a broad dynamic range and it requires no proprietary instruments. This assay, because it is an in-process assay, does not require the stringency of a release assay for the final product. And the last example I've provided here is replication competent lentivirus, or RCL. So the pain points are PCR methods that involve the selection of primers, probes, PCR reaction parameters, PCR reaction agents, can be cumbersome and time consuming and require a lot of training. Also, this type of assay, there could be a problem with PCR inhibition from a matrix of the sample or the pre-analytical process. So the solution here is to use droplet digital PCR, commonly known as ddPCR, instead of qPCR, to partially alleviate the effects of the matrix interference and remove the need for standard calibration. So we have to talk a little bit about stability testing, because stability testing is very different for cell-based therapies. Since cell therapies are what's called living therapeutics, with the final product being viable cells, stability testing is very critical. FDA and other regulatory agencies recommend that product stability studies for cell therapies, including CAR T cells, should include testing to determine product shelf life, as well as in-use stability, which covers a much shorter time, could be hours to days, to establish the duration for which cells can be held under specific condition. So there are two types, right? In-use stability and stability testing to support shelf life. Purpose of in-use stability testing is to establish a period of time during which the product can be used while retaining quality within accepted specification once the container or the bag or the vials or any of those cell vessels that the cells are cryopreserved in. In-use stability is critical for delivering reproducible results for cell therapy products, both fresh as well as cryopreserved. Product stability testing is required to ensure that cell therapy product is at minimum stable for the duration of the clinical trial. And this is a very important criteria. In order to determine the product shelf life, stability indicating assays should be identified through stressed stability sample studies. This is a little more complicated than the standard assays. And these stability indicating assays should be related to functional, phenotypic, and physiochemical properties of the final product. The table here actually comes from the chapter, and you can see examples, stability testing plan for an autologous CAR T product to determine its shelf life. And it lists the attributes, the tests, and the duration of the time. This is just an example, but most sponsors usually have a stability plan in their CMC, which is pre-approved by the FDA. But viable cell count, the cell viability, safety, identity, potency are very important attributes. And you can have any test. These are just some examples of cell counting, sterility assay, identity using flow cytometry, percent of CAR positive and CD3 positive cells, and then potency, which can have any type of assay. This is just mentions the cytokine assay. And on the right side, you can see time points following lot release. It's right after, and then at 3 months, 6 months, 9 months, 12 months, 18 months, 24, and 36. Again, this is just an example, and stability plans can vary from product to product. Next slide, please. So I want to talk a little bit about process analytical technologies, or PAT, also commonly known as in-process testing. And this is defined as a system for analyzing and controlling the manufacturing process through timely measurements of Attributes that are listed here: critical quality attributes, overall performance attributes of raw materials and cellular byproducts. Some people call these intermediates, overall final product quality, and real-time evaluation of the manufacturing process. In simple terms, what we would like to understand is, in process testing, is it allowing for monitoring of the cells as they go through their journey from one unit operation to the next to the next? For example, in a CAR-T workflow, in-process testing can evaluate and confirm cell isolation, cell activation, transduction efficiency, and expansion parameters. As these cells most of the times are in closed automated instruments, we need to understand if they're doing really what they're supposed to be doing. And that is the beauty of in-process testing. You can see an example, again, from the book chapter of technologies and measurements in the path space. And some of them are used routinely, and some of them are still not as common, but becoming common. So as you can see, there is NIR spectroscopy, Raman spectroscopy, fluorescent sensors, refractive index measurements, multi-wave fluorometry, holographic imaging, impedance, turbidity, media component measurement using HPLC, LCMS, Coulter counter for biomass, imaging of the cells, and photometric analysis. And the measurements, if you take a look at them, are really important because they address cell metabolites, viability, osmolarity of the cells. They talk about pH and dissolved oxygen. They also understand compositional changes, amino acids, biomass availability, phenotype of the cell's shape, viability, and then a detailed understanding of media and changes in media components when the media are spent. So process analytical technologies really help us understand if our process is robust and if it is going to meet the requirements to manufacture the product in a safe and efficacious manner. Next slide, please. So we talked about process analytical technologies or in-process testing, and while it is important and necessary, it requires removal of significant number of cells at multiple time points. And as you already figured out, this can create a problem, especially for autologous therapies where the cells are so precious, and the loss of cells in the in-process testing can negatively affect the final dose. In order to avoid this problem, newer advancements such as the use of non-invasive, non-destructive cell analysis technologies have been implemented by many sponsors and manufacturers. Such technologies allow for sample acquisition and monitoring at multiple time points without significant risks of contamination, loss of cells, and risk of failing to meet the final dose requirements. And a very good example of this, which is in the public domain, is the partnership for technology between Bristol Myers Squibb and a company called Ovisio. And they did a project called Automated Cell Monitoring in Real Time and Label Free. So their data has shown that non-invasive, non-destructive technology can provide real-time monitoring of attributes, help with interface of bioreactor and cell therapy processing equipment seamlessly, interface directly with controlled automation for physical and digital connectivity, utilize pre-sterilized single-use products to avoid contamination. And this report is very good because it also shows the data that was generated for CAR-T cells. They also are very valuable in understanding the journey of the cells as they move from one unit operation to the next. Use of such technology, according to the report, enabled BMS scientists to expand their workflow, use a dye-free approach for viable cell counting, conduct media feeding, and monitor critical quality attributes in real time. Another example is the use of sensors to monitor cell health, viability, pH, DO, and other metabolites. The beauty of this system is these sensors are at the surface level of the bags or culture vessels, and they do not come in contact with the cells. On the right side of the slide, you can see examples of sensors. You can also see the Ovisio system attached to a bioreactor. And then the bottom two are actually rocking motion sensors and stir tank bioreactor that can take these type of sensors. Next slide, please. So we would be remiss to not talk about potency assays, right? Potency assays are critical as they measure all aspects of the therapeutic and biological ability of a product. They are necessary part of product characterization, testing, comparability studies, and stability protocols. And in many cases, we have seen that potency assay is not required to initiate early phase clinical studies, but a qualified assay to determine the dose is highly recommended by the regulatory agencies. So on the right side, again, I have taken a table from the chapter, and it describes the common challenges in developing potency assays along with examples that you can see. So to just walk through them, some of the challenges of potency assay development, as many of you are familiar, is inherent variability of starting material, limited lot size, limited material for testing, limited stability, lack of appropriate reference standards, many active ingredients in the product mix, and potential for interference and synergy. And that would be more for gene therapy. So the top 5 are the ones that are challenges for cell therapies. So this is an important aspect. I have listed a reference below if you needed to go have more information on potency assays and specific potency assays as they relate to cell therapies. Next slide, please. So we would be remiss again not to discuss the challenges related to potency assays and issues that some sponsors have faced from a regulatory perspective. We have read this in news as well. And as many of you may be aware, on October 19th of 2022, Alliance for Regenerative Medicine and American Society for Gene and Cell Therapy, ASGCT, organized an all-day working session, including senior FDA staff, cell and gene therapy developers, and academic experts. And this session was focused on development challenges related to potency assay requirements. And on the top, I have actually listed a quote from Dr. Peter Marks. He says, I think there's pretty uniform agreement that one of the key things that has delayed a fair number of approvals over the course of time has been issues related to potency. So the regulatory agencies admit that there is a problem. One of the key takeaways was that the developers also challenged regulators to consider the scientific basis for multiple assays within an assay matrix that yield highly correlated results. And the suggestion was that it may not be necessary to perform potency assays on every stage of the therapeutic cascade, for example, infectivity, expression, activity, but to do it at the late stage of the process. Because, for example, the late stage of the process can only come about if all previous stages were completed successfully. So since we have Mike here, We will probably spend a little bit of time discussing that in the QA part. I've listed some of the other challenges that are faced by many developers and sponsors. There are no standardized potency assays due to the complexities of mechanism of action. In many studies, although characteristics such as migration, proliferation, persistence, and specific phenotypes are linked to a positive clinical outcome for many adaptive cell therapies, only the cytotoxic activity of the effector cells led to a reduction in tumor burden and is therefore considered to be the preferred mode of action for a release assay. And this is an important point because there has to be a connection of what's happening in vitro and in vivo. The interesting fact is potency assays for the two FDA-approved CD19 specific CAR T cell products, Kymriah and Yescarta, measure the percentage of transduced cells and interferon gamma production in response to CD19 expressing tumor cells. The studies have shown that interferon gamma production vary greatly from lot to lot in clinical trials for Kymriah, complicating the correlation of interferon gamma production to product safety or efficacy. And then there are many papers that talk about the fact that interferon gamma secretion measured for effector cell activation, but interferon gamma is not always found to be secreted by cytotoxic lymphocytes. So currently used potency assays, they involve labeling of target cells such as chromium 51, live or dead, antibody staining, for markers, flow cytometry, reporter enzyme assays, cell attachment assays. The underlying physical and biological mechanisms for quantifying drug potency and measuring cell-mediated toxicity differ substantially. And it is important to take all of these into consideration. Next slide, please. So just a little bit about advancements in analytical assay space. For example, artificial intelligence. Unlike other forms of blood cancer, acute myeloid leukemia cannot currently be treated with CAR T therapy since the specific molecular targets are lacking. So an interesting study was done at the University Hospital of Munich and AI Institute of Munich. They just published a paper in Nature Biotech, and they used AI and extensive bioinformatic analysis and came up with two candidates after screening 25,000 potential cell surface molecules. So here is the power of AI. There's also examples of using quality by design with AI techniques to control CQAs of biologics, and this can be adapted to cell therapies. Scientists are already using deep sequencing, omics, single cell sequencing, and other specialized algorithms to improve analytical assays. And then there are ongoing efforts to use AI-based computer vision system into GMP-regulated environment, which can eventually replace subjective and error-prone human operator readings for QC assays. Other examples are machine learning to build predictive models, use of automated sensor chips to improve analytical assays, and use of AI to build feedback loops and connectivity for real-time raw material management. Next slide. This is my last slide. So in summary, what we have discussed, I'm just going to summarize. We need robust and well-characterized analytical methods to ensure safety and efficacy of the final product. Since cell therapies are living therapeutics with cells as the final product, analytical assays need to be designed early in process development stage and subsequently refined to ensure product quality and optimal CQAs. Regulators are okay with a phase-appropriate approach with active risk assessment that can help streamline implementations. New strategies need to be adopted, as I mentioned, to overcome challenges related to turnaround time, cell loss, and human touch points. One thing that we have learned is communication and transparency between developers and FDA is critical so that we do not face the same delays as we have in the past. And lastly, advancements in the analytical assay space, such as AI, machine learning, and bioinformatics, will improve safety, speed, and accuracy, and help with getting these much needed therapies to patients in a timely manner. I want to thank you for your time, and we can go to the Q&A session. Thank you.
All right, thank you, Rupa. And we have quite a few questions. While I'm compiling these, maybe I'll just ask you one that I have. Could you say maybe just a little bit about T-cell mediated assays for cytotoxicity for measuring
Yes.
CAR T potency?
Yeah. So CAR T as an example, right? We mentioned that we're using CAR T as an example for the A-cell project, but it applies to many other T cells. The standard assays currently are the chromium release assays, the radioactive version, the non-radioactive version. They are complex because they require incubation of effector and target cells. Then there is the bioluminescence assays, the standard one being the BLI luciferase. There is impedance assays, there's flow cytometry being the most common. It is important to note that flow cytometry needs to be evaluated critically because all the data we generate is using healthy donors, and then the profiles of the patients do not fit. So it's very important to understand the gating strategy. And then just a word on some alternative assays. There is a very good paper that was published in December of 2021, I believe, by a Sloan Kettering group. And they have talked about alternative assays, which is direct measurements of target cell lysis products such as calcine. They also, of course, mentioned the one that is becoming more and more common, which is cytotoxic assays based on measurement of cytosolic enzymes. Then there is the ELISpot or the enzyme-linked immunospot assays, interferon being common, but granzyme and perforin is also becoming common. So these are some of the traditional assays and advancements in the T-cell mediated cytotoxicity measurements.
Thanks. So we have some good questions from the audience I want to make sure that we get to. First question is about analytical comparability. And are you able to comment on a well-designed strategy to generate sufficient comparative data to permit interpretation of historical batch data in the context of results produced by a revised method? So as your analytical methods change, how do you deal with analytical comparability? And do you do that via retains, or what are the other strategies to employ there?
Yeah, thank you for that. I just actually read a very, very good paper on comparability studies, and also I think there is a narrative by the FDA on comparability studies that has been adopted from biologics to cell therapy. Retains is sometimes the only choice that sponsors and manufacturers have. I think it's very important to create a comprehensive plan for comparability studies, because if they are not accepted by the regulatory agencies, there can be delays. But I've also seen approaches where when people have already decided that they may need comparability studies, they are generating cells in the GMP space and storing those cells to conduct comparability studies. One example is many sponsors or developers of cell therapy may be doing in-house GMP manufacturing, and now they want to go to a CDMO, and comparability studies might be needed. And so they have to set up these parameters very carefully from the data that was already generated and the data that will be generated, and they will use the cells that were generated for this specific purpose. If people don't have the luxury to do that, then it is important to have adequate number of retains to do the comparability study. And the discipline for the comparability study is similar to the discipline that you would use for establishing analytical assays.
Got it. Thank you. Another question, do you run FIO assays, or do you think it's wise to run FIO assays on clinical batches?
Absolutely. absolutely. I have seen time and again that there are many companies who have very strong IP portfolio in the adaptive cell therapy space, or CAR Ts or CAR NKs, who have a little bit of window into understanding the inherent variability of some of the release assays. And they are proactive, and they will start developing very robust FIO assays. And if what their little suspicion or fear does come through, they can go to the FDA, they can show the data generated using the FIO assays and ask for some of those variability in the release assays to be replaced by the much more stronger, consistent, and reproducible FIO assays. And FIO assays a lot of people think that it's a lot of work, but if it is planned carefully and systematically, it doesn't require that much work. The other aspect is FIO assays also give you a window into mechanism of action. So if you are a multi-product company, or you want to continue to improve your product to second generation, third generation, FIO assays with the window into MOA become the foundation for the next step.
Great. So there's a question, I think I'll take this one, if you
Sure.
don't mind, Rupa, about
Please,
stress
please.
stability studies on T cells at higher temperatures when, and the question is essentially, are they representative, or how can you make them representative when these cells are intended to be stored in liquid nitrogen? So it's a good question because, I mean, the good news is these cells are very stable, right, in liquid nitrogen.
Yeah.
The bad news is that makes it hard to conduct a stability study because they're too stable from that perspective. The only real guidance I can offer there is that I've seen examples where you don't necessarily need to go to a full thaw condition or a room temperature condition, and you may want to evaluate various temperatures in a preliminary engineering study as you're trying to understand stability. So you might go to like a -20 C, for example. So hopefully that answer provides some insight.
Mike, they've also done a step-by-step study. So from liquid nitrogen to -80 to -70 to 4 degrees.
Right. Yeah, that's essentially what I was trying to say. So thank you for that clarification.
Of course.
There's a question about a recording. We will post that to our website within a couple of days or a week. One more question about FIO. Can assays addressing critical quality attributes be FIO, or do they need to be release assays? So if something's a critical quality attribute, does that need to be measured with a assay for release, or can you do an FIO?
I have not seen any examples of CQAs tied to FIOs. I'm sure internally people are doing all kinds of things, but in terms of what the regulatory agencies require, is that the release assays, they are kind of the set aside holy assays, and they need to be performed. I think if the FIO assays show better connectivity to the critical quality attributes, I think people have to go to the regulatory agency, show the data, and have a conversation. Mike, I know you have said this many times, and I want to say this as well. My interactions, your interactions, our colleagues' interactions with the FDA have been positive for the most part, right? A lot of people are afraid to go and ask questions to the FDA or regulatory agencies. And what I'd really like to say in this last minute is, please take advantage of the fact that the regulatory agencies, at least now with Peter Marks and the big change that is happening, the number of people that they're going to hire, take advantage, talk to the regulatory agencies, be transparent, be open, be communicative. I have heard from many developers is not doing that actually delayed their process. So Now, I do want to say, at least from experiences that are my personal ones and what I have seen, yes, they don't respond in a timely manner, but I do feel like they want to genuinely help. They understand the intricacies, the complexities, and the uniqueness of cell therapies. And they are sensitive to the issues that the manufacturers and
So
sponsors.
I think the lack of responsiveness that you just mentioned is in part a resource issue. And we know that CBER is going to be bringing on, in excess of 120 new FTEs this year as a result of the PDUFA reauthorization. So this is going to take some time to resolve, but I think we are on our way to resolution.
Yeah.
And I agree with you. You have to talk to the regulators and disclose data.
And don't be afraid.
Some of the feedback we got from the October meeting from the regulators is, these are my words, but don't go on a fishing expedition and ask the regulators, what should we do?
Yeah.
Show some data about your product and explain to them what you think should be done based on the data. And I'll just offer that as a final thought. We're out of time, unfortunately. I know we did get some more questions, but we'll compile those and post the answers in a couple of weeks to our member portal. And we will make a recording of the webinar available on our website, like I said, within the next week or so. Take care, everyone.
Thank you. Bye.